Welcome to the Class 2 of Instrumental Methods of Biomedical Analysis, Infrared Spectroscopy. In the previous class, we discussed about what is IR spectroscopy, different regions of IR spectroscopy, theory and principle of IR spectroscopy, and different vibrational modes of spectroscopy. Today we will discuss about some instrumentation of IR spectroscopy. As we have studied in UV visible spectroscopy, there single beam and double beam spectrophotometer will be used. Here also same single beam and double beam IR spectrophotometer will be used, but only the difference is source of light and detectors will be different. Rest the sample cell, sample holder and monochrometers are same. There are two types of IR spectrophotometer, that is single beam or single monochrometer IR spectroscopy and double beam IR spectroscopy. This is the block diagram of IR spectrophotometer. It consists of source, that is source of light, monochrometers, detectors and the mode of operation, that is recorder. This is the block diagram of double beam spectrophotometer used in IR spectroscopy. It is also known as dispersive spectrophotometer. The difference between the single beam and double beam here is two monochrometers will be used with two additional mirrors will be there, and rest will be the same, that is source of light, sample compartment and reference compartment, and two mirrors will be there, that the beam is going to be collimated and it is fall on the two different monochrometers and then detectors and then recorder. This is the diagram of single beam infrared spectrophotometer. As you can see, a beam of light, it passes directly to the sample and it passes to the mirror, to the prism and to the retromirror. Both the prism and retromirror select the desired wavelength and then it allows to pass to the detector, and detector gives the signals. This is the double beam infrared spectrophotometer. You can see the two beams, that is the reference beam and the sample beam is collimated on the collimating mirrors. Two mirrors are there. This is simple diagram of the double beam infrared spectrophotometer. It is also called as dispersive IR spectrophotometer. Both sample cell and reference cells are different. Two beams is going to be passes directly onto the sample and the reference cell. The source of light, that is C, on the previous slide you have seen, is made to split into two beams, one of which passes through the sample, that is called sample beam, and the reference beam, that is standard we will keep. The double beam arrangement facilitate in measuring difference in intensities between two beam of each wavelength, that is one beam is fall on the sample, another beam is fall on the reference standard, and it is having three slits, that is S1, S2 and S3. One of the two prism monochromator has been replaced by a dual grating. So it means here in the single beam we use prism monochromator, but in double beam you remember always it is used as a grating monochromator, and that is going to be passes to the detector. Detector picks up the signal and amplifies. This amplifies gives the reference beam and the sample beam. This is advanced spectrophotometer that is called Fourier transfer IR spectrophotometer, that is called as FTIR. The interferogram signal which receives by the computer. So here a moving mirror is kept, which is called as a interferometer. FTIR uses interferometer to process the energy sent to the sample. The source of energy passes through a beam splitter where a mirror is placed at 45-degree angle to the incoming radiation. This allows the incoming radiation to pass through but separates into two perpendicular beams. When the two beams meet at the beam splitter, they recombine, but the path length difference, that is wave number, will be different of the two beams, causes both constructive and destructive interference. The combined beam containing this interference pattern is called as interferogram. The interferogram contains all radiative energy coming from the source and has a wide range of wavelength. The interferogram generated by combining these two beams is oriented towards the sample by the beam splitter. As it passes through the sample simultaneously absorbs all frequencies that is normally found in the infrared spectrum. This is the modified IR spectrophotometer. Thank you very much.